So lymphatic malformations, or LMs, have heterogeneous phenotypes. The majority of them are due to somatic variants in the PIK3CA, with hotspot variants E542K, E545K, and H1047R, leading to the hyperactivation of the PI3 kinase AKT-mTOR pathway. This results in increased cell proliferation and decreased angiopoietin 2. PI3 kinase signaling is also known to regulate transcription, translation, cell migration, metabolism, and survival. mTOR also regulates protein expression and degradation via the proteasome and VEGF-adherent expression. Thus, we were interested in how PI3 kinase hyperactivation affects protein expression. So by immunofluorescence, or IF, patient-derived endothelial cells had increased VEGF-adherent and CD31 expression, shown in red, with white arrows, when compared to control cells, which have sunny side-up appearance, as these junctional proteins are located at the surface, cell surface, in contrast to abnormal cytoplasmic accumulation in patient-derived cells. And by FACS analysis, we see that this increased cytoplasmic expression in patient cells is correlated with the reduced VEGF-adherent and CD31 surface expression, as shown with the red arrows when compared to the controls with green arrows. Thus, we hypothesized that PI3 kinase hyperactivation leads to proteasome defects in LMs. In order to test our hypothesis, we used lymphatic malformation endothelial cells, or LMECs, isolated from patients with confirmed PIK3CA hotspot variants, and human dermal lymphatic endothelial cells, or HDLECs, as controls. We engineered HDLECs to express hotspot variants by lentiviral transduction, and control cells were HDLECs transduced with an antivector or wild-type PIK3CA. Cells were then analyzed by IF for VEGF-adherent CD31 and the proteasome marker PSMB5, and transduced cells were also stained with phospho-AKT to confirm successful transduction. In all images, mean signal intensity was determined with MHA and normalized to the cell number and compared to control cells. Statistical analysis was performed using the one-way ANOVA with a post-hoc test. Next, we used the transgenic LM mouse model, where PIK3CA with H1047R variant is specifically induced in the lymphatic endothelium by the ear, topical with 4-hydroxytamoxifen. The LM was allowed to develop for 4 to 5 weeks, then ears were harvested, and whole mount or transverse sections of the ears were stained for VEGF-adherent CD31 or PSMB5 and the lymphatic marker LIVE1. Images were quantified for lymphatic vascular density or signal intensity, and results normalized to vessel area. Statistical analysis was done with a Mann-Whitney test. NMECs with hotspot variants specifically increased VEGF-adherent and CD31 and PSMB5, which co-localizes with VEGF-adherent, shown in yellow. The increase in PSMB5 is actually a readout of the proteasome insufficiency. That is, proteasome levels increased to address the defect in protein degradation. Next, we evaluated the engineered HDLECs relative to controls. All 3 PIK3CA variants significantly increased AKT activity, confirming a successful transduction, and shown in red in the top panel. And similar to LMECs, all engineered HDLECs had significantly increased VEGF-adherent and CD31 expression, both in green, and PSMB5 in red in the bottom panel. To evaluate the relationship between AKT activity and VEGF-adherent, a regression analysis was done, and it shows a significant correlation between AKT activity and VEGF-adherent levels. The LM mouse model had abnormally increased lymphatic vessels associated with defective VEGF-adherent expression pattern. At the lymphatic front, on the left, the capillaries bottom-like VEGF-adherent pattern in white arrows in controls was lost. Instead, a zipper-like prolactin duct phenotype was observed in the lymphatic plexus on the right. Normal valves in green arrows were seen in controls but lost in the transgenic model. And the transverse sections of the ear revealed ectatic vessels, shown in yellow arrows, with significant increase of VEGF-adherent CD31 expression correlating with increased PSMB5. To conclude, we showed that PIK3CA hotspot variants are associated with increased weak adherence CD31 proteasome in both LMECs and the engineered HDLX, as well as the transgenic mouse model. We demonstrated that pathogenic hyperactivation of PI3 kinase pathway downstream the hotspot variants lead to proteasome insufficiency in LMECs, in NMs. Finally, I would like to thank all people who make this work possible. Dr. Junwoo, my PI, Professor Kerry Schober, lab members, Ben, Sam, and Mason, and our funding sources. I'm ready for questions. Thank you for your attention.
Hi, Nick Avicula from Brussels, Belgium. Very nice presentation. I was just wondering, because we tried to do the HDLX as well for different studies, so you use the lentiviral vector. So what's your transduction efficiency? And do you know what's the level of expression of the mutations, the mutant allele?
So the efficiency here in this case is, we have to optimize a little bit to hit about 90% of efficiency, I would say. We had a green control, a GFP control, to see the cells that were transfected. Once we started to check for the cells with IF or the PAKT, actually to check if they were actually having a hyperactivated pathway, most of them were actually hyperactivated. So the transduction was quite successful.
So you don't select them with FACS or something with the gene?
I do not do a selection. There's no.
The mixture that you use?
Yes, and actually this would sometimes sort of mimic the mosaicism that we see in patients. But in this case, most of the cells were actually successfully transduced.
So do you know what's the level of expression of the mutant allele compared to?
I can answer that. It turns out really high expressors die, so we don't get it, and so the level is very low. So we routinely go to PAKT to look at the levels. It's very hard to detect PIK3CA. There's not really good antibodies. We've done RT-PCR, and so it's a subtle, maybe a one-fold increase. Everything else dies. So we get a lot of death initially, and then we get what comes out of that.
Because when you highly express, usually they die. The cells don't like.
Exactly.
Okay.
This is
Very
a
good. Thank you.
PGK promoter, which is much lower.
Hi, Brian Sisk from Washington University. This is a really fascinating area of interest. And when you think about the clinical applications, it could be that you need efforts to help the proteasome to work better and make the cells happier, or it could be you make the proteasomes work even less well and cause the cells to die. Which do you think is the right approach if we try to translate this clinically in the future? Do we want the cells to be happier and healthier, or do we want them to die off?
Okay, this is a very interesting question because this is the next step of the study. You're very welcome to attend to my next talk that would address this. And
I'll take my check in the mail.
But I can give you a hint actually, because of the proteasome actually here would help to get rid of the pathological cells actually, so we want to make it work even less.
I do have actually a follow-up comment for that. I know that about 2 years ago, and I hate to point out Dr. Junwoo, who really gave a great talk, and I remember 2 years ago on the promise of proteasome inhibitor. Could we follow up on that and what's going on with that and this particular research that we just heard about? No, the use of proteasome inhibitor in possibly clinical setting or clinical trial with now the more evidence that you have of the importance of proteasomes?
I'm sorry,
Okay.
Hi, Twee.
Yes.
It's actually his talk in the next session.
Okay.
It's our update on that work. And June and I also published a review of all the BTZ uses now in Frontiers of Cardiovascular Medicine. Oh, pharmacology. I'm sorry. I have 2 different papers out in Frontiers. So it's actually published the use of BTZ, at least not in our patients, but the safety profile.
Okay, appreciate it. And with that, we are just out of time. So thank you so much.